Hey, welcome to today's video. My name is Rochelle and I am your microbiology tutor. Today's video, we are going to learn about microbial growth and the control of microbial growth. We're going to learn, we're going to study together, and I'm going to test your knowledge to see if what you are learning, what you are studying is sticking. All right, without further ado, let's get started. Binary vision. The cells elongate to approximately twice their original length and then form a partition that constricts the cell into two daughter cells. And here you see an illustration. The parent cell, the DNA duplicates, and then the cytoplasm divides, it elongates, and then eventually creates two daughter cells. An example is E. coli. Here. Next we have generation time. It is the time that is required for one cell to separate from to form two cells. Budding division. Now, bacterial cells that divide as a result of unequal cell growth forms a completely new daughter cell with the mother remaining. Here we have two examples. We have budding in yeast, and then we have hydra budding. So with budding in yeast, we have the nucleus, we have our parent cell, we have the forming of the bud, then the cytoplasm divides with the nucleus, a duplicated nucleus in that bud. And then we have the parent, and then the chain of yeast cells form, and the daughter yeast is formed. Here we have the hydra budding, where we have the parent cell, the parent hydra, and then there's the budding that's developing, and then we have a new bud, and then a new hydra. Biofilms. Now, biofilm is a polysaccharide matrix that contains bacteria. So we're going to put that here. Polysaccharide matrix containing bacteria and nutrients. Bacteria cells. Specifically, we have platonic growth, which means that the microbial cells can grow in suspension. They don't have to be attached to anything. Sessile growth is microbial cells that grow and are attached to surfaces. Some more information about biofilms. Well, it is very important because, one, it prevents harmful chemicals entering into to harm the cells. So it's the prevention of harmful chemicals. Two, we have it's a barrier to protect it from outside invaders. So it's a barrier, protection against invaders. Like protease. And then three, it prevents the cells from being washed away. So prevents cells from being washed away. Okay, on to growth phases. Oops, a little bit. We have a lag phase, exponential growth phase, stationary phase, and the death phase. Right here on the right, we see a graph where the log phase, which is the first one here, number 1, it is the growth that begins to happen after a period of time. So this is like the start. Essential exponential growth phase is the doubling of the growing cells at a regular interval, and we see that exponential growth, and that's 2. What happened to my number 1? Let's clean that up a little bit. Number 1. All right. Next, we have stationary phase. And now it's after the essential growth phase when it ceases due to the lack of essential nutrients or there's an increase of waste product. It's accumulated. That's phase 3. Oh man, these numbers. Okay. We are going to 3. And then we have the death phase where there is no net increase or decrease in cells. And we see a decline here on the graph. And that's 4. 1, 2, 3, and our 4th last one. Okay, I think it's time. Time to do a quiz and test your knowledge. First one, bacteria cells that divide as a result of unequal cell growth forms a completely new daughter cell with the mother remaining. Now, what you can do is pick the correct answer, of course, and also put your answers in the description box, in the comments section. And then I can go ahead and check them. It's A, binary fission, or B, budding division. Second one, elongating. Well, that was number 1. Number 2, cells elongate to approximately twice their original length and then form a partition that constricts the cell into 2 daughter cells. Is it A, binary fission, or B, budding division? Three, regarding biofilms, microbial cells that grow in suspension. Is it platonic growth or sessile growth? A or B? Four, still regarding biofilms, microbial cells that grow attached to a surface. Is it platonic growth or sessile growth? A or B? Place your answers in the comment section. Okay, this is a mix and match game. So we have 1, there is no net increase or decrease in cell numbers. Is it A, B, C, or D? 2, essential growth ceases due to lack of essential nutrients or waste products accumulate. Is it A, B, C, or D? 3, doubling of growing cells at a regular interval. So is it A, B, C, or D? We have growth, last one, number 4. Growth begins to happen after a period of time. Is it A, B, C, or D? Again, place your answers in the comment section. Oops. Let's do a little mathematics for microbial, for bacterial growth. So we have our first equation where n is our final number of bacteria, and the initial n here is the initial number of bacteria. Oops. Let's erase that, make that look a little bit better. And small m in here is the number of generations. So it's number of generations. So you'll be provided, probably if you're on your exam, a chart where you have the cells, the number of cells, and here you'll have time, and you'll have a line, let's say here, and you have 2 points. This one would be your initial. Oh, well, I shouldn't put initial. I should put it would be in, oops. And then you'll have another point here, which would be your final number. And then you would just, well, you can plug in and then end up finding n. But in order to find n, another way is here with this equation. 1.3, and you have the log of n, and you have n here, and minus the log of the initial n. You do the calculations, and you'll end up getting your n. So that's another way that you can find your n. Another way, we have g equals t over n, where the n, or n, we have the n, which is the number of generation. We have t is the time, the duration time, and that is how long it takes to for the bacterium to double. So we have the number of generation, and then t is the duration time. And then g, we have here, it is the generation time. Oops, I wrote over that. Here. So we have 1, 2, 3 equations that we can use when finding generation time, and that is very important. So in order to find n, you can use this equation. If you have the final number of bacteria, or you have the initial number, then you can do the math and find n. Or you can go here, use this one to find n, plug in your duration time, plug in your number of generation from this equation, and then you'll find your generation time. Okay, next subject, still in math, instantaneous growth. There we go. Okay. That would equal our little k. So instantaneous growth rate is k, equaling 0.693 over g. Well, we know that g equals t over n, and above we already discussed. So you would need to gather information for T, gather information for N to get the G, plug it in, do the math, and you'll have your instantaneous growth rate. Here we have chemostat. Let's adjust the screen. Put that in the center. Okay, we have chemostat. It's a device wherein both the growth rate and the cell density can be controlled. And I need to put it in right there. Can be controlled independently. So for example, let's show you, you have your device and the medium. The media's in there. You inoculate it with cells. They begin to increase. There's also a way where cells can be removed. And then we also have in here limiting nutrients. Oops. Nutrients. And that can be carbon, or it can be like a nitrogen source. So when we have an increase in our cells, cells can be removed, but there is limiting nutrients in the medium, which can be carbon or a nitrogen source, then there is a decrease in the specific growth. Growth rate. Now there are two factors that govern the specific growth rate. And one is dilution rate, D, where D equals F over V, where F is the flow rate. So that's the flow rate. It's supposed to be W. Flow rate. And then the V is the volume, the culture volume. Oops. The second one is the concentration of the limiting nutrient. So the idea is that there should be a balance or a steady state between 1 and 2, between the dilution rate D and the concentration of limiting nutrients. And when you have I guess the right mix, then you have a steady state. So to be more precise, you have the dynamic condition in which the cells are continuously flowing, continuously growing, and continuously moving out of the device. And in order to do that, you need the right dilution rate and which is the flow rate over culture volume, and you need the right amount of concentration of limiting nutrients, which is either like a, for example, carbon or the nitrogen source. So steady state, you want continual growth and continual removal of cells, so you can have a balance. So the purpose is having enough cells in the exponential phase to use in lab. That's one. Two, can study microbial ecology and evolution. And then three, it is used for direct enrichment and isolation of bacteria from nature. Next topic is media. Here's some definitions. Defined media. There are precise amounts of pure inorganic or organic chemicals to distilled water. Complex media are made up of digests of microbials, animals or microbial animal or plant products. Yeah, the enrichment medium is used to culture nutritionally demanding microbes like pathogens. We have selective medium. It inhibits the growth of some microorganisms but not others. And differential medium has an indicator in it which allows the medium to change colors when a particular metabolic reaction occurs during growth. Quiz time, another mix and match. And this one's about the medium. So, one, are made up of digest of microbial, animal, or plant products. And then here are your options below or on to the side. Two, inhibits the growth of some microorganisms but not others. Three, there are precise amounts of pure inorganic or organic chemicals to distilled water. Four, has indicator, which allows the medium to change colors when a particular metabolic reaction occurs during growth. Five, used to culture nutritionally demanding microbes such as pathogens. So this is a mix and match, and you'll choose whether it's A, B, C, D, or E. And then you can place your answers in the comment section for this quiz. Let's see. Next up, we have temperature. Temperature can be used to control the growth of microbes, or it can increase the growth, or it can be optimal for a particular group of microbes. And we're going to discuss these groups of microbes below. So we have psychrophiles. They are cold loving, and their temperature can range from -10 to 15 degrees Celsius, and their optimum temperature is 10 degrees to 15 degrees Celsius. We have, well, this is supposed to be a Y right there, Psychrobacter arcticus, and it is, you'll find these microbes in ocean deep or ocean depth or the polar regions. We have mesophiles, and their range is 10 degrees to 45 degrees Celsius. No, that looks like a 95, and I don't want to write that. 5 degrees Celsius, and the optimum growth temperature is 25 to 40. 35 to 40 degrees Celsius. We have E. coli as an example, Salmonella species, Lactobacillus species, and they are found in animal bodies or in food spoilage. Continuing on, we have thermophiles. Thermophiles, I want to change my color. Okay, thermophiles, their range is from 40 degrees to 70 degrees Celsius, and their optimum temperature is 50 to 60 degrees Celsius. And we have Thermus aquaticus, and they grow in, you'll find thermophiles in hot tap water, the sunlit soil, hot springs, and organic compost piles. Okay. Next we have psychotrophs, and their range is from 0 to 30 degrees Celsius, and their optimum growth is 20 to 30 degrees Celsius. And we see Listeria monocytogenes, and the environment that they would grow is in the refrigerator, and you'll see food spoilage and just in the natural environment. Hyperthermophiles, their temperature range is 65 degrees to 110 degrees Celsius, and their optimum temperature of growth is 85 degrees to 100 degrees Celsius. And the example that I have here for the microbe is Pyrobolus pyrodictium. And you would find these hyperthermophiles in hot springs, volcanic activity, sulfur metabolic activity, and the deep sea hydrothermal vents. In order for you to remember this, I would like to show you mind mapping, micro mind mapping. So I'm going to choose a different color here. So what you can do is put the name of the group, for example, hyperthermophiles, and then you're going to stem off with temperature range, and then stem off for the optimum temperature. Then you're going to give an example of bacteria, and then you're going to give examples of their habitats. And you'll do that for each one. Hyperthermophiles, psychotrophs, what else did I have here? Mesophiles, psychrophiles, thermophiles. You do that for each one, and you just practice it. And then when you have that down, then sit down and just recite it. Hyperthermophiles, they have a range of 65 to 110, but their optimum temperature of growth is 85 to 100 degrees Celsius. The microbe associated with one of the microbes associated with hyperthermophiles is Pyrobolus, or you can name some other ones. Their habitats are hot springs, sulfur metabolic activity. They are found in deep sea hydrothermal vents. So you'll be able to recite that once you have your micro mind map down. So that's a good way to practice and study. Here are 3 questions. Feel free to answer and put them in the comments section. So, question number 1. What type of microbes are able to tolerate acidic environments? 2. What is the optimum temperature of pathogenic bacteria? 3, which organism can live in hypertonic environments? So these questions will allow you to dig a little bit deeper in your textbook and do a little more study. Okay, on to our next topic. I'm going to center here. And this is microbial growth and pH. So I know everyone are familiar with pH ranging from 1 to 14, where 1 is closer or is more acidic than we have 7, where it's neutral. And then we have 14, which is alkalinic. Here are some examples of bacteria that can thrive in acidic environments. Oops. Delete that and change the color there. So we have bacteria that can thrive in acidic environments. We have Thiobacillus acidophilus and Lactobacillus acidophilus. And you would find Lactobacillus in your mouth, in your intestines, and in vagina, the female reproductive body part. Next, we have pH of 7, or ranging from 6.5 to 7.5, which is neutral. We have E. coli, the Salmonella species, and Staphylococci. And you would find these bacteria in humans and in soil in nature. Lastly, we have alkalinity, which is in the 12, 13, 14 pH scale. We have Vibrio cholerae, which is a bacterium that can cause cholera. And cholera stems from you can get cholera from contaminated water where feces are in the water. Then we have nitro. A natronobacterium is another example here. And that's for alkalinity. Yeast. So mold and yeast have a pH levels ranging from 5 to 6. Now, when you're in lab, you want to, of course, grow bacteria, and some bacteria can produce. You're in the lab, and you want to grow bacteria, and some bacteria will produce acid, but you don't want them to produce acid. So in order, this is the objective, to stop the growth, there are buffers in place in the medium. And here are 3 buffers that are used in media, and it's peptones, amino acids, and phosphate salts. Oh, phosphate salt. Oops. There. Our next topic is aerobes and anaerobes. This one's going to take quite a bit of explaining, so I have a link which would be in the description box of this video. It's a 30-minute video where I go in depth in describing aerobes and the anaerobes and the environments that allow them to grow or not to grow. So you can watch that. The next one I have is another video that you can watch, and that will be in the description box as well. And it's the control of microbial growth. And I go over moist heat sterilization, boiling, and autoclaving. And once you get that information down, then you can do another right here, mind mapping. So it's a mind mapping, or again, you put your, your main word here, and then it stems from different informations that you've learned through the video, through your reading, through your studying. And then once you are able to recreate this without using your notes, the micro mind mapping, then you are able to, you can sit and recite. Okay, boiling can be used to kill endospores, but it's going to take more than 20 hours for endospores to be killed. It can kill bacterial pathogens, viruses, fungi, and spores. Autoclaving, okay? Autoclaving can be used to kill endospores, viruses. You'll need a high temperature and a high pressure. Autoclaving is used in healthcare to sterilize instruments, dressings, and solutions. So once you learn about boiling and autoclaving, then you'll do your micro mind mapping. Repeat that a few times until you have it down, and then here, explain it in your own words. Okay, here's a question. What moist heat sterilization method is best used to kill endospores in 15 minutes? You can place your answer in the comment section. Pasteurization. Pasteur is known for being the father of pasteurization. He kind of like figured out how to prevent spoilage and by using mild heating. So pasteurization is the purpose of it. The intent of it is to eliminate pathogenic microbes, to lower microbial number. What we see here is that pasteurization can kill thermodermic bacteria with heating. Examples are used here for pasteurization is ice cream, beer, or yogurt. We have high temperature short time, where there's 72 degrees Celsius. That's the temperature needed for 15 seconds to kill pathogens and lower the total amount of bacteria. Again, we're going to do some micro mind mapping. You have your main words. So here we have Pasteur, the scientist known for pasteurization. We have some key points: spoilage, pasteurization, and mild heating. And then from there, each one of these, you would add information to it. Then repeat this mind mapping, and then be able to recite it to yourself or to someone else. Again, do that with pasteurization, and then high temperature, short time. And that's the mind mapping. Question here on the bottom: what is the intent of pasteurization? All right. Dry heat. Again, all of this information is in my video, and the link will be in the description box. So we have flaming and hot air sterilization. Flaming is the direct flames, the use of direct flames. For instance, when you're in your lab, you're inoculating loop where you are sterilizing it over a Bunsen burner. So we have incineration. Incineration can be used for two reasons: for sterilization and for disposing items such as paper cups, dressing, and bags. With hot air, let's see. Let's center this a little bit here. Okay, so with hot air sterilization, the equipment that is used is the oven. So the temperature would have to be at 170 degrees Celsius for 2 hours in order to sterilize the item in the oven. Again, we're going to do some micro mind mapping. You have your main word, and then you're going to stem off to your your main word, and then your subwords, and then just continue adding and building. You have your examples, and then you will be able to recite all of this information. That it's flaming is used in either direct or in incineration. With direct, you can use it in your lab with your inoculating loop, putting it over the Bunsen burner. That's direct. Then you have incineration, where you can sterilize or dispose of items such as dressings, bags, or paper cups. Then we have hot air, and the instrument used is the oven. Here's the temperature, the time, and it's used in sterilization. Question: what is the temperature and the length of time for hot air sterilization? Go ahead and place your answer in the comment section. Now, the last two topics, we have sterilizing radiation. We're using ionized radiation and non-ionized radiation. I have a video that will be in the description box that you can view and study, and then in that video I test your knowledge. And I do the same thing for the terms and definitions of control of microbial growth. In that video, it will also be in the description box, and I go over the terms and definitions. We study it together, and then I test your knowledge in that same video. I also have in the micro workbook that I gave last week that's available. I'll have that link in the description box as well, where you can download your workbook and follow along in the videos. So that is the end of this lesson. I am so glad you were able to view and study, and I was able to test your knowledge. I hope you did well, and I will see you in the next video. All right, bye.